Biochemistry of enzymes and vitamins. Vitamins are vital micronutrients that cannot be synthesized endogenously or an insufficient amount, and the principal means by which we get vitamins is through our diet. Vitamins can classify as water soluble or fat soluble. The fat soluble vitamins include vitamins A, D, E, and K. Fat soluble vitamins play integral roles in a multitude of physiological processes such as vision, bone health, immune function, and coagulation. This review discusses the biochemistry, transport, and roles of these vitamins, highlighting deficiency syndrome and potential toxicities. Despite structural differences between fat-soluble vitamins, they are absorbed and transported similarly due to their low solubility and hydrophilic media. The body absorbs fat-soluble vitamins into newly forming micelles in the small intestine. Micelles are lipid clusters that contain hydrophobic groups internally and hydrophilic groups externally. This process relies on the secretion of bile and pancreatic enzymes. After absorption into enterocytes, fat-soluble vitamins become packaged into chylomicrons, which then get secreted into the lymphatic system before entering the bloodstream. Chylomicrons are metabolized by lipoprotein lipase, which causes the release of fat-soluble vitamins into tissues for use and storage. Many enzymes are simple proteins consisting entirely of one or more amino acid chains. Other enzymes contain a non-protein component called a cofactor that is necessary for the enzyme's proper functioning. There are two types of cofactors: inorganic ions, example zinc or CO or ions, and organic molecules known as coenzymes. Most coenzymes or vitamins are derived from vitamins. Vitamins are organic compounds that are essential in a very small trace amount for the maintenance of a normal metabolism. They generally cannot be synthesized at adequate levels by the body, and most be contained in the diet. The absence or shortage of vitamins may result in a vitamins deficiency disease. In the first half of the 20th century, a major focus of biochemistry was the identification, isolations, and characterizations of vitamins. Despite accumulating evidence that people needed more than just carbohydrates, fats, and proteins in their diets for normal growth and health, it was not until the early 1900s that research established the need for trace nutrients in the diet. The substrate binds to the enzyme primarily through hydrogen bonding and other electrostatic interactions. The induced fit model says that enzyme can undergo conformational change when binding substrates. Enzymes exhibit varying degrees of substrate specificity. Initially, an increase in substrate concentrations increases the rate of an enzyme's catalyzed reaction. As the enzyme molecules become saturated with a substrate, this increase in reaction rate levels off. The rate of an enzyme's catalyzed reaction increases with an increase in the concentration of the enzyme. At low Temperatures, an increase in temperature increases the rate of enzymes catalyze reaction at higher temperatures. The protein will denature. Enzymes have optimum pH ranges. Many enzymes are simple proteins consisting entirely of one or more amino acid chains. Other enzymes contain a non-protein component called a cofactor that is necessary for the enzyme's proper functioning. There are two types of cofactors. Inorganic ions, for example, zinc or CO, or ions and organic molecules known as coenzymes.